This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 831, recorded on November 18th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
This is clinical update number 89. Looks like we're going to hit 100, Daniel.
Yeah, it's unfortunate, right? I mean, going into this a lot could have been avoided, but yeah, this is tough. All right, well, before I do my quote and get started, I wanted to take a moment and share the passing of Dr. Scott Hammer. I don't know if you knew, but Scott passed away last night. Dr. Hammer was the head of the Division of Infectious Diseases at Columbia from 1999 to 2019, when Dr. Magda Sobieszczyk stepped into that role. And he was the head of ID when I came to Columbia, and he I think was a special individual. He was one of those rare individuals who, despite being brilliant, world renowned, making such incredible contributions throughout the world with his work, he was just humble. He was kind. He was the kind of guy you could just walk in and start talking, and then he would realize, like, oh, do we have an appointment? Do we have something we're supposed to be addressing? But just like just a nice guy. And I think Vincent, you knew Scott as well.
Oh yes, I did. He used to be involved in teaching of the microbiology to medical students, and I saw him all the time. And then I did an interview with him on TWiV, actually, quite a few years ago. We'll have to dig that up and put it in the show notes.
Oh yeah, I think actually that would be nice.
Talked about HIV, and I hadn't heard it, but then again, I only go up to Columbia a few days a week, so. Yeah. It's too bad. I'm really sorry. He was really a nice guy and a good clinician. He ran a lot of HIV/AIDS clinical trials, right?
Yeah. Whenever I would travel in sub-Saharan Africa, everyone knew Scott Hammer. He was really just a tremendous amount.
Too bad.
So, all right, let's get right to our quotation. Go forward with courage. When you're in doubt, be still and wait. When doubt no longer exists for you, then go forward with courage. So long as mists envelop you, be still. Be still until the sunlight pours through and dispels the mists, as it surely will. Then act with courage. And this is Chief White Eagle, the Ponca chief. And I thought this was appropriate. This is Native American Heritage Month. A friend of mine, Kenny Cord, is actually a member of the Shinnecock tribe here on Long Island. We went to high school together, and I've had a chance to talk with him over the last couple of years here. Nice to talk to him right after the vaccines had rolled out, because I was reaching out, checking in, seeing how that was going, and they really did a great job. And it's sort of the way to do it, right? When the community themselves mobilize and they're doing the vaccination as opposed to other people stepping in. Unfortunately, the Shinnecock Indian Nation's 75th annual powwow was canceled this year. Kenny had invited me. I'm hoping for a rain check in the future. But I think there's a lot of wisdom here, and I think this quotation can be applied in so many ways. I think we've talked about moving forward with randomized controlled trials, how there needs to be a certain amount of patience. But I think also a lot of people are not sure about the vaccines. There's a need for the development of vaccine confidence. And I think it's all right for people to ask questions. And I thought it was appropriate to put this in because this is going to be the last episode that drops before those conversations around the turkey table. Mm-hmm. And it's going to be tough, I know, at times. So I'm sort of remembering something that I'm going to modify a little that my grandmother Anne had in her house. And it was, I'm going to wish everyone the serenity to accept the things you cannot change, the courage to change the things that you can, and the wisdom to know the difference. I think the biggest thing as families come together is be ready to listen. I think these conversations are important, but be ready to learn as much as you expect to teach. So my friend Kenny actually runs a non-for-profit out there, actually, Hoop School, helping these individuals, actually athletes from less advantaged populations, move forward with school and things like that. So, all right, what is the update? Case numbers are rising. Here in New York, the 7-day average is back to around 6,000 cases per day. But back in April 2020, prior to vaccines, when we had this daily case number, we were seeing over 400 deaths per day. Currently, same case number, the daily death number is about 30. So just sort of compare what happens when COVID sweeps through an unvaccinated population versus a highly vaccinated population. So when I speak to urgent care providers, the number of positive tests is rapidly rising. So we expect these numbers to go up, but our local hospitals are actually relatively quiet in terms of COVID admissions. So I think I think there's something really educational here. And we'll be touching several times on the difference between a vaccinated and an unvaccinated individual getting infected. We're getting the thumbs up on small family and friend gatherings for the upcoming holiday from some of our top public health experts. People could probably guess who I'm referring to. But we're also being warned that the case numbers are high. People should still be making smart decisions, wearing masks when they're out in indoor settings, large groups of people. But we'll be talking a little bit about steps that people can be doing so that in those family friend gatherings, those masks can be off, right? We're not going to be sitting around the turkey table with masks on, like feeding turkey under the mask or something. All right, important updates and dates. We heard from the FDA that on November 30th, the advisory committee will meet to discuss the available data supporting the use of molnupiravir to treat mild to moderate coronavirus disease 2019, COVID-19. This meeting was scheduled as soon as possible, I'm quoting here. Following the submission of the EUA request by the company, this timeline allows for the FDA to thoroughly evaluate the data and information submitted in the EUA request before the meeting and to be prepared for a robust public discussion with the advisory committee members. So I think that's exciting news. We have not yet heard a date when the Pfizer Paxlovid medication will be discussed by the FDA, where it has been requested. We anticipate that will be some date in December. There was a little bit of a twist in the EUA, and Vincent, and maybe you'll weigh in on this because I'm going to bring this up a few times going forward. They are requesting approval for Paxlovid for the treatment of only unvaccinated people. What are your thoughts?
I don't know why they would restrict it, because if you have a vaccinated person who gets infected and they're at risk, you might want to treat them, right?
Yeah, I think that this will come up in a little bit when I talk about one of our monoclonal studies in vaccinated individuals. There certainly are vaccinated individuals who have a number of risk factors that put them at that, let's say, 10% or higher risk of ending up in hospital. I would love to have access to this medication in that population. My hope is either the FDA says just let's not restrict access here, but maybe that's just going to be a temporary thing. But we did hear news, right, that the U.S. government is investing billions of dollars, over 10 million doses of this, courses of this medication. So hopefully we're going to be able to move forward and not just restrict it. All right. Yes.
Why do you think they would do that? Do you think they can't make enough and that might be part of it?
So my thought about why they might be restricting is that, when you do these studies, you want to power them, right? So you don't have to have thousands of people. So you could say, all right, I want a population anticipated, let's say 8 to 10% chance of ending up in hospital or death. Right. And then I want to reduce that by a certain percent. If you start doing a study with vaccinated lower risk people, you're going to need a much larger number of people to participate to get to that endpoint. There was the Racaniello drug, which actually enrolled a lower risk population, and they couldn't reach the power, right? So they actually abandoned their ongoing trial. So that might be one of the issues. People might say, you didn't study this in vaccinated people. But I think it's going to be tough. Here's someone who has active viral replication. I'm going to want, as a clinician, to use an antiviral.
Of course.
All right. Children are at risk for COVID. I think that's very clear. And wearing a mask is less traumatic for a child than being hospitalized. We now know about 25% of the new cases in this country are actually in children. But on the good side of this, by last count, there have been almost 3 million children that had been vaccinated in this 5 to 11 year range. Talking to parents, watching for any reports. If anything, the children seem to be having much less reactogenicity than the adults. Maybe they're just tougher than we are, but yeah, I feel like they want to have a little bit. I was talking to one of my infectious disease colleagues just yesterday, and his son went and got the shot and then had to go swimming. And he's like, "Daddy, I think he calls his father Papa. Papa, I think my arm might be a little sore, but that was about it." So,
but Daniel, do you think a 5-year-old has already a fully mature immune system?
well, I wouldn't say fully matured. I think this is interesting, right? We have different sort of stages when and that's really why we broke down vaccines into these. Right at about, we'll say 4 or 5, there tends to be one sort of development, then there's another development. So there are sort of developmental milestones. So they're not fully mature at this point, but they have gone through. And so, it'll be a slightly different situation with the under 5.
Maybe that's part of the reason they don't have as much reactogenicity.
Yeah, actually, that might be part of it. It may also be smaller dose, right? It's the 10 versus the 30 versus the 100 with Moderna. All right. Testing. Now, this is going to be, I think, we spend a bit of time here on testing, isolation, quarantine, because a lot of people have been asking me a lot of questions. I think we need a refresher because I am hoping there will be a lot of testing around the holidays. Testing really is a critical piece to making the holidays safer. So I keep getting questions like, what's going to happen, right? If you do all these tests, a positive test is going to come in. And what do you do? So we're going to reinforce the guidance on quarantine versus isolation. So remember, you quarantine when you may have been exposed to the virus, right? These are those ships sitting out there for 40 days off of Italy waiting to come in because there may have been an exposure. Isolate for the infected. So this is for people who've been infected, even if they don't have symptoms. So we'll start with isolation, because isolation is simple. One isolates for 10 days from either the first positive test or the date of symptom onset. We've talked a little bit that I think the science suggests that we might eventually have different rules for vaccinated people, might be able to shorten this, but we're not there right now. Right now it's 10 days. Quarantine, this gets a little more complicated. First, people who are fully vaccinated do not need to quarantine after contact with someone who has had COVID-19 unless this vaccinated individual has symptoms. However, it's not just free and clear. A fully vaccinated person, it's recommended that they get a test 5 to 7 days after that exposure, even if they don't have symptoms. We recommend that they're really careful with wearing that mask indoors in public for about 14 days following exposure, or at least until after that 5 to 7 day test is negative. Now, it's different for the unvaccinated. This came up today in one of our urgent care settings. For the unvaccinated, the quarantine lasts for 14 days from the last contact with a person who has COVID and is in the isolation period. So this is that time that we think they're infectious. But the CDC allows a little bit of flexibility, and some states have kind of gone down one of two different roads. One is quarantine can be shortened to 10 days without testing. So that's the New York State option. So New York, it's easy. Isolation for the infected, 10 days. Quarantine for 10 days, no test out option. Now, other states, this is another option the CDC puts out, after day 7 of quarantine, if you have a negative test, you can shorten that quarantine and test out. But that's a state by state, and that's not something we do in New York. But let me go through a little bit of like how complicated and challenging this could get. And this was a case that came up today. We have a mother and father. The mother's unvaccinated. The father is vaccinated. There's reasons why the mother has decided that to delay a little bit. She's not going to get vaccinated. She just was waiting to get vaccinated. But the son tests positive this morning. Now, what are we going to do? The son is going to need to isolate for the infected for 10 days. But mom is going to continue to take care of the son. The father's vaccinated, right? So he's kind of free and clear. Maybe he'll do that test 5 to 7 days if he's following the recommendations. But mom stays, takes care of the son for 10 days. And that's the last day of her exposure. And then she's supposed to quarantine for another 10 days, and she can't test out in New York. So it's 20 days.
Wow.
This is tough, right? This is why everyone speeds. This is why no one listens to the rules. It's really tough. I mean, the percent of people that really follow all these rules is small. And this is part of this test to stay approach, the idea of daily rapid antigen testing, because people are not following these rules.
So why doesn't New York State recognize that and allow tests to stay?
So it's currently under consideration, right? We have a number of pilot programs in different places. We talked about Utah. We talked about Massachusetts. Looking forward, right? It seems like it will make sense at some point because I don't think we can sustain this, right? I mean, COVID's here to stay. Are we going to continue to every time our child gets, infected, lose 20 days of our life or, the 10 days? It's not
I mean, Daniel, look, as you said, the hospitalization is way down. We don't need to do this anymore.
Vincent, I feel like you're disagreeing with the CDC and our local Department of Health, but no,
it's not the first time. Wouldn't be the first time.
Okay. All right. What about those serology tests, right? I feel like I have to keep repeating. So we've talked about the PCR, the antigen, and don't worry, we'll get back to that. I was on a call earlier this week with a physician, actually, who was considering getting a booster shot. He had COVID. He had some issues after his second dose of the original mRNA vaccine. And we're having a conversation. We're going back and forth. I feel like it's all going really well. Mm-hmm. And then he says, maybe I should just go get my antibodies checked and then make a decision based on those levels. And that's when the buzzer went off. And I said, oh, you were doing so well up until that point. So the article, SARS-CoV-2 reinfection associates with unstable housing and occurs in the presence of antibodies, was published in CID. Clinical infectious disease. And the authors found no significant differences between the SARS-CoV-2 receptor binding domain, nucleocapsid IgG levels, ability to neutralize in a pseudovirus assay between the convalescent and the reinfection groups. So just, people keep looking for this. You want to be able to, and on an individual level, we're not able to see this population level, right? There is some sort of large association, but we cannot make clinical decisions. And the FDA has been pretty clear about warning people that is not something you should be doing as a clinician. All right, active vaccination, never miss an opportunity to vaccinate. Vaccines, the jabs that keep you from getting sick, sort of point out what the vaccines are supposed to be doing. And, we talk about COVID all the time, but I want to remind everyone, our public service, go out there and get those flu shots. Do not forget about your children as well. In the MMWR, there was the publication Influenza Vaccinations During the COVID-19 Pandemic, 11 U.S. jurisdictions, September through December 2020. And overall, the influenza vaccine administration was 9% higher than average if you look back at 2018, 2019, except for when you look at our children. So it declined among children aged 6 to 23 months by 13.9% and by 11.9% in those 2 to 4. So don't forget about the children. And I think that, Vincent, you may be aware, we're starting to get some flu outbreaks. We've got a bunch going on in New Mexico, a university, I think Michigan, name them. Sorry about that. But it's not your fault. These things happen. So, we don't want to have a twin-demic, which is what people have talked about. And you know what? When the masks come off, not only are we going to see flu, but I also want to keep reminding people that COVID is not the only thing that we need to be thinking about. There was an MMWR entitled National Surveillance for Acute Flaccid Myelitis, United States, 2018 through 2020. And I suspect you've seen this, Vincent. I think this is a research area you and Amy are focused on. But for our listeners, acute flaccid myelitis, AFM, is a recognized complication of certain viral infections that predominantly affects previously healthy children and can progress rapidly, leading to respiratory insufficiency and permanent paralysis. I sort of think it as somewhat similar to a modern polio type syndrome. So this may be associated with enterovirus D68. And we've been seeing these biennial peaks, 2014, 2016, 2018. But then per this report, in 2020, we didn't see it. But time will tell when the masks come off, when we see this next surge. Just a reminder, terrible pathogens out there. We've been a little bit spared and focused just on COVID, but just sort of want to make sure people realize that we still need to focus on all these other areas of critical research, not just COVID. All right. In anticipation of the upcoming holidays, many states are making boosters an option for anyone 18 years of age and older. I will say we're not seeing safety concerns. Third doses tend to give similar reactogenicity as the second. But if one had an issue with that first or second dose, we encourage people to have a discussion with their physician. And if you don't have a physician, connect with a physician and have that discussion. Passive vaccination, remember monoclonals for post-exposure prophylaxis, early treatment, high-risk individuals. Let's move into the period of detectable viral replication, the viral symptom phase, the time for monitoring and monoclonals, not the time for antibiotics, not the time for steroids. I was on a call this week with CityMD talking to their urgent care providers, just sort of reinforcing what we know. Eventually, this is where we're going to have our oral medications, we hope. So just a little review, and I think to reinforce in the coming weeks, the issue of timing, right? So we talked on a previous episode about the molnupiravir MOVE out study. So the out is the outpatients. That was where molnupiravir reduced the risk of hospitalization and death by 50% if given within the first 5 days. But I want to let people know there also was a MOVE in study. And this was the molnupiravir inpatient study. And that actually was stopped, really a futility issue. The issue here, and I think we've tried to reinforce this, is by the second week, you've missed the window. It's too late for antivirals. Paxlovid, we talked about that last week. That was an 89% reduction in the endpoint of hospitalization or deaths if given within the first 3 days, an 85% reduction if given within the first 5. So again, just focused on timing as these become available. Just like Merck, it was announced that Pfizer has signed a voluntary license agreement that will allow qualified generic medicine manufacturers to make their medication in 95 countries throughout the world. So I think that's fantastic, right? No one's safe until everyone is safe. We've got to keep our eye on the world. Now, here gets back to a little bit of my issue and a little bit of our discussion about Paxlovid. What about vaccinated people that get COVID post-vaccination? And so the paper, Monoclonal Antibody Treatment of Breakthrough COVID-19 in fully vaccinated individuals with high-risk comorbidities became available. It's published in JID. Now, here the authors reported on a cohort of 1,395 persons. And just some important details, mean age was 54.3 years. 60% of these were female. The median BMI was 30.7. And these are individuals who developed COVID-19 more than 14 days after full vaccination. Now, the first bit, 107 of these 1,395, or 7.7% of these individuals who developed infection post-vaccination required hospitalization. Now, hospitalization was associated with a number of medical comorbidities, but monoclonal antibody treatment was significantly associated with a lower risk of hospitalization, an odds ratio of 0.227. So about a 77% reduction in going on to require hospitalization if you got monoclonals. But then they have this wonderful table and go through a little bit more detail, because what about the number needed to treat? Well, the number needed to treat is really impacted by who you're treating. So if you treat individuals, the lowest risk patient, right? This is, your BMI is 26, but you're in your 20s and you have to treat, number needed to treat was 225. But once you start moving forward into individuals that have a number of high-risk medical conditions, then the number needed to treat was only between 3 and 8. And once you got up to multiple high-risk conditions, the number needed to treat to keep someone out of the hospital was only 4. So they did a monoclonal antibody screening score, and they actually have this in Table 2. And I reached out to the corresponding author earlier today because they labeled it Table 3, and I couldn't find Table 3. I guess, I just found Table 2. But they're going to fix that. But it is great. If you spend a little time looking at Table 2, you can really see that, if a person has even a single one of these scores, they're going to end up with a number needed to treat of 43. But once you get into several, you're really 3-4 people number needed to treat. So people who have a BMI greater than 35, diabetes, chronic kidney disease, greater than age 65, chronic respiratory disease, these individuals, you do not need to treat very many before you're keeping somebody out of the hospital. I think what's really important about this paper is it reinforces that if you are an individual who has high risk features, vaccination is going to significantly reduce your risk of ending up in the hospital, but there's still a risk. And here we're seeing, so, we often get a little bit complacent, I think, like, oh, you're 80 years old, but you've been double vaxxed. Now maybe you've been triple vaxxed. But if a person has a risk of ending up in the hospital that's 10% or higher, we still want to go ahead and make sure we do whatever it takes, what hoops we have to jump through, get them access to therapeutics. All right. And I'm going to jump all the way to the tail here. We often hear, right, a lot of questions about the long-term risks of vaccines. I don't know if we talk, as much. Well, we certainly do on TWiV, talk about the long-term impacts of acute COVID, COVID that lasts more than 2 weeks and turns into a post-acute sequelae of COVID. We've talked a lot about adults, but this week, the first child was enrolled in the NIH-supported long-term study of children with COVID. Now, this study will track up to 1,000 children and young adults who previously tested positive for COVID-19 and evaluate the impact of COVID-19 on their physical and mental health over 3 years. Now, this is part of the Recovery Initiative, trying to better understand the long-term consequences of SARS-CoV-2. Unfortunately, we are seeing a significant number of children that are not better after 2 weeks. And they're actually going to have the NIH Clinical Center recruiting children aged 3 to 21, and the Children's National Hospital in Washington, D.C. will recruit children aging from birth to 21 years. So we're even seeing this in really young individuals. All right. I don't want to go too long because I know this is going to drop right before the holidays, but just want to close by saying no one is safe until everyone is safe. And throughout the months of November, December, and January, donations made to Parasites Without Borders will be doubled. They'll be matched. They'll be given to Microbe TV up to our goal of $40,000 that we're hoping to contribute to Microbe TV to help them with their efforts.
Daniel, if you get more than say 20,000, because you're going to add the other 20, you'll still accept it, right?
We'll still accept it. We have a very, as Vincent knows, a very cozy relationship with Microbe TV. So we may even support them a little more than that $40,000.
Very good. Love it. All right, time for some email questions for Daniel. You can send yours to daniel@microbe.tv. Pamela writes, weeks ago, in response to a question from a young mother, Dr. Griffin responded that his wife should breastfeed their child. Not Dr. Griffin's wife, but you understand. Okay.
I understand. I got it.
What I was hoping to hear was if there is research that addresses how a nursing mother's milk is impacted by getting a vaccination. Our daughter believes she should not get vaccinated until she stops nursing. I know they are saying pregnant women should get vaccinated, but this is about the next stage of motherhood.
Yeah. No, actually, we recommend that nursing mothers get vaccinated. We actually recommend that mothers get vaccinated before they stop nursing. And part of it is the ability to provide a passive immunization to your child. There clearly are antibodies in breast milk. So, I certainly hear all these concerns. Oh, I want to wait until I'm no longer pregnant. I want to wait until I'm done nursing. We actually are encouraging women who are breastfeeding to go ahead and get vaccinated so that they can actually then protect their children.
Mark writes, I fear By the time this question reaches you, it will be too late for any response you might have. However, your answer might be helpful to others. My fully vaccinated 31-year-old brother tested positive twice for SARS-CoV-2 by rapid antigen test yesterday after developing COVID-like symptoms, headache, sore throat, congestion. Went to the ER at Kaiser Hospital in Oakland last night after experiencing what he described as chest tightness. At my advice, because he has chronic kidney disease, he asked about monoclonal antibody therapy. They began administering the Regeneron cocktail, but they had to stop because he had what he described to me as an anaphylactic response. Eventually, they sent him home because they deemed his symptoms mild or moderate and his blood oxygen levels were fine. He's due to follow up with an ID specialist. My questions for you are several. First, how often does this sort of reaction occur in response to monoclonals? Second, and more important, would it be worth trying a different cocktail if one were available, or would he be likely to have the same reaction? Would it be too late for him to receive another infusion, say tomorrow, 3 or 4 days after symptom onset, if that were possible? And finally, if monoclonals are out of the question because of his response to the first treatment, are there any alternatives available for someone in his situation?
Yeah, so a lot of questions there and really good ones. So let's start off first with the safety profile of the monoclonals. When we look at the published studies, as well as our experience when we ran the largest post-EUA bamlanivimab trial, we are seeing people have, I'm going to call them intolerance issues, about 1 or 2%. It's not really anaphylactic. It's more of an anaphylactoid type reaction. We see it very rarely with the monoclonals because these are not humanized. They're not a mouse chimera, and they're not a mouse antibody that was modified. These are fully human antibodies. So about 1 to 2 people per 1,000 have issues tolerating them. So they're incredibly well tolerated. Occasionally we'll see the heart rate might go up a little bit like that, something. But no, this sounds like one of those individuals, and this does happen, who did not tolerate the therapy. Then it becomes a risk benefit. Does someone try to switch to a different monoclonal platform? And we honestly don't know. So you sort of want to make a risk benefit here. How much of that did they get before the response? We're talking about numbers needed to treat in our study before. So if they had a reaction to Regeneron, do you want to look at giving one of the other products? This is all going to be sort of in the realm of clinical judgment and a little bit challenging.
So, Daniel, what's the window? If you days after symptom onset, what's the longest you should go before you get the monoclonals?
Yeah, so our EUAs define a 10-day window in getting those monoclonals. But clearly time matters, right? You get it within the first 3 days. We're seeing almost no progression to hospitalization. You move into the first 6 days and you actually start seeing some, I'm going to call it breakthrough. Once you get into that 7 through 10, about half as effective as if you got it in the first 6 days. So there's clearly a progression over time. And I think we've talked about the importance of timing with the monoclonals. And one of the issues with a lot of the bureaucracy that introduces delays into what is a time-sensitive therapeutic.
Sleepless in the suburbs writes, I live in a suburban area in a red county. Our town's conservative school board has made masking and post-exposure quarantines optional this year. My 10-year-old son was exposed to COVID at a school a little over 2 weeks ago. I wanted to quarantine him at home. Right word, right, Daniel?
Yes. Quarantine for the exposed.
At home for 2 weeks post-exposure, but my employer would not allow me to work remotely to stay home with him because his quarantine was optional. I reluctantly continued to send my son to school. On his 14th day post-exposure, he told me he had a sore throat and stuffy nose. I gave him a Binax Now home test, which was negative. I thought he might be faking to get out of school, but because of his somewhat recent exposure, I kept him home, got him a PCR test at the local drugstore. His test was positive, and he's since developed a cough. He definitely has COVID, so I've lost my faith a little in rapid antigen tests. I got a Pfizer booster a month ago, so I'm hoping I'll be protected. However, his dad, my husband, is unvaccinated and opposed to getting our son vaccinated. Once my son recovers, how long will immunity likely last? If I got our son vaccinated, it would likely destroy my marriage and result in divorce. Now that my son will have some natural immunity, I'm wondering if it's worth blowing up my marriage to get him vaccinated.
Okay, wow. So there's a lot in there, and let's start off with let's try to restore your faith in testing. So let's think about the pre-test probability. I know we've talked about this before. So your son has an exposure. Now he starts to have a sore throat, starts to have symptoms. So I'm already thinking, ooh, it sounds like worrisome for COVID. Sounds like this is most likely COVID. So you go ahead and you do the rapid test. And I think this will be helpful to think things through. We've always talked about the idea that people in general, start to test positive before symptom onset. But everyone is different. Some people start to have symptoms. And I think we talked about this last week. We had a woman who had symptoms, started Thursday, got a PCR Friday, was negative, symptoms continued. Then she got the rapid that was then positive, right? So she didn't lose faith in PCR, nor should we. But timing and frequency really matters here. If you go into it with a 90% likelihood, sounds like your child has COVID, a negative test, you're still 50% likely it's COVID. So you want to go ahead and you want to repeat the test the next day. That's why a lot of these tests come in 2-packs. You don't want to just trust one test. You want to trust a second test. Now, the PCR was really a second test. So yeah, your son does have COVID. It makes sense. You've got it confirmed by the test. If you went ahead and did a rapid test that next day, it would probably be positive. So think about multiple tests. If it doesn't make sense, and we do this clinically all the time, I've had patients who come in, we do a PCR, it's negative, it doesn't make sense. We repeat it, then it's positive, right? So we don't lose faith in our tests. We just have to understand that frequency and repetition if it doesn't make sense. The other thing, what about protection, right? So you are about a 30% chance of ending up testing positive despite vaccination because you're in the house. This is a really significant exposure. Your husband, who's unvaccinated, does not have that same protection, so has about a 40% chance of ending up testing positive in the next 2 weeks, which you could say 40% means 60% he won't, right? So hopefully he won't, because that might have a negative impact on the marriage. But what about your son going forward? One of the concerns we have about children is they do not seem to develop a consistent protection after, I'll say, virus or infection-induced immunity. So in that case, we would recommend getting your son vaccinated. So, have a discussion. Just keep having a discussion. I don't think you need to get divorced over this, but I think it's become really, really clear. We are seeing surges in children getting hospitalized. I hope and I expect your son to do quite well with this infection, but COVID's here to stay. It's really a question of what's going to happen the next time your son gets exposed.
Well, Daniel, we know that probably he only needs one dose, right, after recovering from infection, right?
It's hard to know, Vincent. I know you say that, but we haven't really seen the studies on that, right? Yeah. So we've looked at, and I'll talk about that study where we looked at adults, right, who were infected, and they got one dose of vaccine versus 2 doses of vaccine. And it was only the 2 doses that reached statistical significance for reducing the chance of them going on to get infected. So I don't know. I don't know if 1 dose is enough. I think it is worth studying.
I'm just trying to save the marriage. Isn't it horrible that a health issue, which is really cut and clear cut, right, should be a contentious one in a household? It's just unfortunate.
Yeah. Are you wearing seatbelts and putting your child in a child seat in the car, right? I mean, how many marriages were lost when seatbelts were introduced? No, it is tough. I mean, and is a middle ground to go ahead after the infection and at least do 1 vaccine? I mean, that seems like a middle ground. So I think that's worth thinking about.
All right. So our last one is all about testing. So this is coincidental. Jamie writes, as another lowly layperson, 72, with sarcoidosis in my lungs, I really appreciate the generally clear explanation of COVID issues that Daniel provides. That said, I still lose the thread sometimes. Case in point was the discussion about the use of CT values and various test results in assessing COVID status. I heard the following. PCR tests do not distinguish between dead remnant pieces of virus and active ones, so a large proportion of positive tests are actually measuring residual dead virus particles in people who are no longer a risk to others. Next, rapid tests are 96% effective in detecting active virus. And third, only about 1 in 1,000 viral replications results in a viable virus. So what does a CT value actually mean? I am completely confused about what conclusions can be drawn in regards to the actual meaning of a positive test of either kind. Could Daniel explain probably again in detail the differences between PCR and rapid tests with a particular focus on false positives?
Okay, certainly. So actually, you're doing great, right? So, and I also like the way you started off with a little bit of the flattering, because I appreciate that. I'm getting a little sensitive here. 2 years in, um, so here's what we know, right? And this story is built over time. And again, I always talk about everyone's a little bit different, right? So you want to be careful when you look at a particular individual. We know from our case control studies that between 2 days prior to symptom onset in general, out to about 10 days afterwards, but really 7 days afterwards is when we're seeing almost all of the transmission, right? So if it was your exposure was to the person 4 days or more before they had symptom onset or tested positive, or your exposure is 10 days after them having symptom onset or testing positive, you have no increased risk of testing positive from that exposure, right? I think we presented that in one of our prior ones. But what is going on with the CT values, and what is going on with our testing during that period? So initially, there is an upslope. With the PCRs, we can often detect the virus, let's say 3 days before symptom onset. The antigen tests start to turn positive about 2 days before, sort of coincident with when we're seeing contact tracing. The PCR continues to stay positive in some individuals for weeks, right? Clearly positive still at about 14 days in most individuals, a lot of individuals. The antigen test, though, starts to turn positive, starts to turn negative at about day 11, 12, 13. So we're seeing this correlation. Now, the CT values, when you're picking it up early, you're going to have to run that PCR many, many times before you pick it up, might be in the high 30s. Then you're going to start picking it up in the teens during that infectious transmissible period. And then you're going to actually start having to run that machine out past 30 cycles once you get down past day 10. So that's sort of putting this all together. Does that seem reasonable, Vincent? You want to jump?
Yeah, I think so. But I think also for Jamie, if he listens to what you discussed previously in this episode where you explained it also, I think that will help him. Yeah. Okay. Or her. Okay. That's COVID-19 clinical update number 89 with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you so much. And everyone, be safe with Thanksgiving coming up. Go ahead, do those tests, but don't just do one, do two.